All right, let's pick up where we left off in class today, because I left off with a bunch of math, but this is math that you've seen before. As I already explained, for transporters are a different kind of enzyme, and I want you to look at them that way. So everything that you're learning about E, S, and P can be applied to SN, S out, and T. Okay, and all we have right here is exactly what we've talked about in this unit already, but we're just changing the labels. And that means that if we have the same 4 steps and the same 6 arrows between them, and we get them down to the same 3 arrows, all the math is the same, and we're just changing out where the labels are. Just realize that the main thing that you have to identify is which direction you're transporting in. The substrate concentration in is your, if you're transporting in, the substrate concentration in is going to be your product. You're going to be from the outside in. Where you're starting is where you are substituting for the S. And that's the case where we have this. Notice that this is written from out to in being transported, and you just flip the labels the other way if you're talking about from in to out. If you do this and you have the same equations, you have the same graphs. And so you have this exact same thing as going on before. Sometimes to distinguish between the two cases, we say instead of Km, we say Kt. If you look carefully at this, you have to squint to even see the Kt, and then S becomes S out in the case where you're doing inward transport. Okay? Everything else is the same. The one thing that we introduce now, though, is a way to talk about the overall delta G of the equation. Because instead of being inherent to the reaction that we're talking about, as it is in an enzyme, in a transporter we can actually calculate the delta G depending on the concentrations inside and out. This is that equation that I told you we were going to put in. That's physical chemistry equation. Again, we'll talk about the why in the physical chemistry class. Right now we're just going to talk about the what, which means that you can tell about the delta G of any transport if you take, and this doesn't depend on in or out, it just means you are moving from initial concentration C1 to final concentration C2. You put the final concentration on top. And all that you do is, this should look familiar because it really is just a version of delta G equals RT natural log of K. The K that we're putting in is going to be the final concentration over the initial. Just make sure you keep those things straight, and this should be easy to take care of. If you have them in the same units, the units cancel out and it becomes a dimensionless thing inside the natural log. So for example, if whatever your concentration units are, if you are moving from a place where you have 100 times less concentration to 100 times more, you're taking natural log of 100, which you multiply that out with R and T, and I believe this is at room temperature. I'd always have to specify whether it's room temperature or 37 degrees Celsius because it matters to the equation. But if you just plug in 100 natural log and by regular RT, then you end up with a positive delta G of transport, because it is not spontaneous to move something into a place that's more crowded. It's more spontaneous to move the other way around. But if you just flip it around, that number becomes negative, and you end up with a spontaneous negative delta G of transport. This is all very similar to what you've done before, but getting it into the new context is going to be the trick. So different glucose transporters, for example, have different Kt values, and you can think about why would a glucose transporter in this type of cell need to bind glucose more effectively. Kt is essentially binding the glucose. Remember, we have to put a little asterisk on that, but the same kind of asterisk goes on this as goes on Km. And the lower the Kt, the better the binding. So look up and down on this and say to yourself, what kind of organs have the tightest binding glucose transporters? And if you look up and down, I see the smallest numbers for the smallest numbers are at the bottom. And so you have heart, skeletal muscle, and kidney. You have a lot of the heart ones are at the bottom. The brain one is sort of, there's a brain one that's 1.4, that's GLUT3, and you see that's very different from the ubiquitous one. The brain one binds more tightly than the ubiquitous one that's at the top. So kind of the average glucose transporter is going to be 3, and this is millimolar. But the brain binds tighter, heart binds tighter. Things that need energy more tend to bind more tightly. So let's look at that basal one. The GLUT1 is sort of the average, and the other things that you can think about how they deviate from average. The brain needing more energy, it makes sense that it binds more tightly to glucose. So that is all you do to figure out what's going on if glucose is. And it's a relatively simple equation, right? RT natural log ratio. But you might think about, what about the case where you have a solute that's charged? Glucose is not charged, and so we only have to worry about concentrations. If the solute is charged, then the difference in charge between the inside and outside matters. And so we have to add another term to our equation. We have a concentration imbalance. We also have an electrical potential imbalance. So it matters what the other charged molecules are. The good news is we can measure this imbalance of all charged molecules by just measuring the voltage across the membrane. And so that's why the voltage matters for these things. So the voltage is measured in millivolts, and at equilibrium, if you don't have an imbalance, you don't have a voltage. So if you have a perfectly balanced pluses and minuses on both sides of the membrane, then you don't have a voltage and this term disappears. If your solute is uncharged, it doesn't respond to a difference in voltage, so you don't have to use this part of the equation. It's only when you have an imbalance and the solute is charged, because the charge will influence the energy of moving the solute in or out. This is a good example of when you have a voltage imbalance. If you allow the salts to move across the membrane, they will eventually come to equilibrium, where you have an equilibrium not necessarily of concentrations, but an equilibrium of charges when it comes to this particular measure. So if a solute is charged, the good news is the part we're adding to our equation is pretty simple. It's just multiplying 3 things together. We give them fancy symbols because we don't see these anywhere else, so all the easy symbols are already taken care of. But the fancy symbols, it's really just 3 things multiplied together that you add to your delta G for transport of a charged solute across the membrane. So you add in Z fancy F delta psi. And if you look at that, Z is just the charge on the ion. If you have something that's more charged, it's going to be more responsive to the voltage difference. So it's just 1, 2, or 3, depending on the number. If it's +1, you put in a +1 there. Fancy F is a Faraday constant. That's going to be given right there, and that's also given on a test. And then the only thing that's even remotely interesting mathematically is the delta psi, which is the voltage. It's the transmembrane electrical potential expressed in volts. So the one thing to look out for is make sure your units are canceling out right here, especially with Faraday constant versus if you have a potential in millivolts. Normally the delta psi is 50 to 200 millivolts, and you see I have the volts there. Be able to move back and forth between those, cancel those out. It's the same story for almost all biochem math. And notice also that the normal delta psi is the charge is more negative inside the cell. The one thing I will say about this equation, when you're using it, you can be very careful about signs. You can say, oh, I know I have a plus here, and I know that my charge is more negative, so I'm going to have a minus there. And that's one way to do it. But the way I like to do it is I actually like to do it intuitively, which is to say, okay, my delta G is going to be moving. I'm moving into the cell, so I know it's more negative inside the cell. I'm moving a positive ion inside the cell, so I know my overall term is going to be negative. Delta G for moving something positive into a more negative environment, it's going to be more stable. More stable means negative delta G. if you can figure things out that way, you can just plug that in for this term right here and not worry about, oh, do my negatives cancel out? You know, what's the real standardization for this and all that. Basically, do the homework problems, think it through, choose whether you want to do it mathematically or you want to do it my way, which is saying what is going to be spontaneous for this kind of charge. And that leads you to the worksheet and the homework. And you can now complete all of the homework 6B, which includes Chapter 11 problems, which were included in homework 6B. Now, I will not actually stop the recording yet because I have one more story to tell that actually ties in with membranes. I have specific investigations of 3 enzymes. So I'm going to throw the first one in with the rest of these because we've talked about the last thing that I mentioned, the inside of the cell is more negatively charged, right? Well, why is that? It's because of this example, the sodium-potassium ATPase, which is a proton, well, it's not a proton, it is a pump for sodiums and potassiums. The insides of cells are consistently different than the outsides of cells. If that's the case, the way that they maintain that has to be something that you put energy into. And guess what? If it's energy that you're putting into, it's probably going to be that you're burning ATP to do it, because that's the energy currency of the cell. And that's exactly what you have. For most normal operating cells, including neurons, for which this process is hugely important, you have the sodium-potassium ATPase is an antiporter that does active transport. That means it's not just opening a door and passively letting things run down the gradient, passively letting things run to where the negative delta G is going to be. What you have is it's putting in ATP. ATP is giving it the negative delta G that it needs, and then it's doing a reaction that actually has positive delta G by itself. But because it's tied ATP hydrolysis to this reaction, you end up with an overall negative delta G, which means that you are paying for a positive delta G with the negative delta G. This kind of thinking is what biochemistry math is for. It's a little bit hard to say in words. It's actually easier for you to calculate. So I'm not going to try to say it in words too much, but I'm going to give you the overview and then let you calculate how it works out. So the thing that it does is it moves 2 ions against their concentration gradient. So it's moving sodium from a higher concentration to a lower concentration. Notice that that is and I should be very careful when I'm using words. Sometimes I say things wrong. Let me go from the slide to say things right. It's actually moving sodium and potassium, both. The sodium is moving out of the cell and the potassium is moving into the cell. And so the thing is, potassium moving into the cell is straightforward. You're moving something into a more negative environment. But the sodium moving out of the cell is actually going to cost you energy because you are moving something into a more concentrated environment and you're moving it into a more positive environment. So both of these reactions actually cost energy. It's just that the sodium costs energy both in terms of charge and in concentration, whereas the potassium costs energy only in terms of the concentration. This is how the cells set up this imbalance, and they do these 2 positive delta G reactions by coupling them to a third reaction that is negative delta G, that is the ATP hydrolysis. ATP hydrolysis is actually a number that was included in previous homeworks. It's a hugely important number, but if you just hydrolyze enough ATP, you get enough negative delta G to run these positive delta G reactions, and then you end up with a cell that has sort of a stored up energy in the sense of it has a constant thing that you can count on. More potassium inside, less sodium inside, and more negative inside. You can see that the more negative inside comes from the other 2, because if you're moving 2 positive things in all the time and 3 positive things out, you're having an imbalance of charge. So this is really cool, but this is the advanced application of all these of all these equations. And this is kind of the hardest math that you're probably going to get on the test. There's other hard math too, but this is as hard as it gets, okay? Neurons, of course, depend on this a lot. If you have something that depends on sodiums and potassiums moving in and out of the cell, then it's going to depend on this. The good news is once this protein does all the work and sets up the general state of firing neurons, then you can basically make everything else run off of this. Everything else can be passive transport once you set up this active transport. And so that means if you're thinking about sodium channels, we've already talked about those, potassium channels, both of those are actually passive transport because this one has done all the active transport work to set up those weird things where you have more stuff inside than out. In fact, it's a lot of work that this does because neurons depend on it. They need to keep this running all the time, and 25% of ATP consumption, when you're not moving it for using it for moving muscles, 25% of all resting ATP is for this one protein. And the process of thinking and studying actually depends on this protein doing the work, using the ATP that you get from sugar to be able to turn that into the work of moving ions back and forth. It's all hanging together by this protein. This is a quote I like. A typical cell in your body has 1 billion ATP molecules in it, and if you calculate how fast those are drained by this one protein, in 2 minutes, every single one of them will have been hydrolyzed and another billion will have taken their place. Every day, if you add this up, you produce and use up a volume of ATP equivalent to about half your body weight. Now, technically, this is a little bit overstated because you're really talking about bonds being broken and reformed. A lot of that ATP is actually reused, but it's a valid point if you think it phrased a little bit weird, okay? If you feel the warmth of your skin, that's your ATP at work because that depends on ATP being hydrolyzed to be able to help. Talk about this being one of the major energetic costs of the cell. If you have the cell stressed by its environmental being polluted in some way, in fact, if the oceans are acidified by increasing CO2 levels, you might expect it to show up in how much energy they have to devote to this one protein. And in fact, that's the argument of this paper right here. They're looking at, under acidified conditions, they're seeing that larvae that are growing have to actually allocate their energy differently. In more acidic conditions caused by more CO2, they're going to have to spend more energy to pump the sodium potassium in and out. And they don't have a choice, they have to do that. And so the numbers right here, the main numbers right here, I don't want to get into the biological details, except I want to show you ATPase activity is shown in gray. And if you especially look, if you look as they grow, if you look at A from left to right, you can see that there are different times where the gray will move up and down depending on the life stage. But in all those cases, if you compare the control, normal conditions, to the CO2 conditions, in all of those cases, the gray is bigger under CO2 conditions. That means they are depending more on sodium potassium ATPase activity. They're running this one protein more because it's harder for them to maintain their voltage gradients that they need to do everything else based on the stress that they have from the increased acid from the CO2. You can see in part B, you have a big difference. They sort of add up all the ages between 6 and 8, and you can see there's almost a doubling in one case, or more than a doubling at age 8 of the 20 for the gray wedge becomes a 45 for the gray wedge. That's a huge difference, and that implies that something is really out of balance with the organism. And this could be a location of one of the stresses. The metabolic energy budget has been thrown out of whack by the increased CO2 and acid that comes with it. And so we can talk about the structure of the pumps. It's really cool, and the one thing I wanted to say is that I think I misspoke earlier. I said a proton pump at one point, you might have noticed. Well, there's a reason for that. Because there's proton pumps that look just like the sodium-potassium ATPase pumps. It's a very complex structure, but it's very similar to simpler bacterial structures that don't pump sodium-potassium ions, they pump protons. This is a very important protein, and how it works is very important, and that means if it works this way, we can have an identical kind of problem where we give you and we ask you, how does a proton pump work? Well, we give you the same numbers, and it's the same energy equations as for the ions, and it might be kind of simpler, so you might see some things about this, and that would be why. If it's an ATPase, if it's a pump that's running on ATP, we can use these equations to talk about it. And in fact, if you inhibit it, you have an important drug. There's this molecule right here, which is a weird molecule, right? If you look at it, it's called wabane, and it comes from foxglove. If you've ever heard about it, digitalis is actually, it's like the Latin name for foxglove, and the extract from foxglove is literally called digitalis. And they noticed a long time ago it helped with heart failure. So it's toxic under some cases, but some toxins, when you are in a bad way, they will actually kick your body back into the right configuration. That's exactly what this does. It actually will block the sodium-potassium ATPase, and it will block that pump. If you have too much of it, it's really bad. But if your heart has stopped, blocking the sodium-potassium ATPase will actually allow the backup generator, the backup system, to kick in, which is a sodium-calcium antiporter. And the thing about that is that gets calcium into the cell, which starts the muscle contraction. So it took us a long time to figure out exactly what's going on. But yes, it turns out that this toxin actually inhibits this very important protein. But it's good because that's how it works, and it results in the heart getting kick-started from the calcium being put into it. Kind of a complex biology, but that's what we get to at the end of this chapter. We get to these kind of stories where you can put all these things together. And it's very cool. The other thing that you can add to this story is you can add the concept of an ionophore. An ionophore is something that is like a siderophore. A siderophore is a small molecule that binds iron. An ionophore will just bind ions that are charged and of a certain size. The thing about ionophores is they can bind things like sodium and potassium. And potassium is the kind of thing that's supposed to be inside the cell, right? And you're counting on the membrane to keep that inside the cell. It has a concentration gradient where if it could, it would flow right out of the cell. If you have an ionophore, you actually disrupt that because the ionophore, look at this ionophore right here, has a charged inside, but it has hydrophobic sort of methylvaline type groups on the outside. And that means that what it can do is it can act like a channel. It can bind the ion, and once it binds the ion and allows it to pass through the membrane, then the ion will move down its concentration gradient, and all the potassium will flow out of the cell without doing anything. Basically, that will kill the cell. The good news is your cells have defenses against this, but bacteria don't. And so you have some bacterial antibiotics that work exactly this way. They are hydrophobic on the outside donuts with the hydrophilic inside. The ion binds into the hydrophilic inside, and this allows them to move down their concentration gradient so that then they can move to where all the potassium from this high concentration potassium to low concentration potassium. They'll do that very naturally and happily, and that will disrupt the voltage of the cell, and that will kill the cell. So there's if you think about it, the ionophore is acting like a channel. What if we could we make a real like fake channel out of some other molecule? And it turns out that this molecule, amphotericin B, which I believe was discovered as an antibiotic, will actually disrupt this whole system. And so this is where we get to a kind of complex problem, but we want to walk you through what this does. Amphotericin B does not directly bind the ions, but it binds the membrane. And what it does is when it binds the membrane, it forms something like that glucose transporter that we showed you. Glucose transporter was 4 helices that were arranged in a circle that made a hole in the membrane. This will do something like that, where it will arrange itself in a circle, and it will make a hole in the membrane that will be polar. You see all those OH groups on that lipid. We'll talk about the lipids in a while. Basically, when we get to that, you can see that the amphotericin B will basically act like a glucose transporter, except it will let ions through. And what that does, they showed how it worked with this figure, which I kind of like, even though it's a little complex. They have the normal situation on the left side, where you have the purple protons, and then you have green potassiums. Under the normal situation, you have the protons being pumped out, you have the potassium, the protons will flow out, the potassium will flow in, and that's just normal growth type stuff. Remember, this is all in the context of a living cell, so you have a normal voltage difference negative on the inside. If you block the channel letting the potassium back in, then you will actually have a problem where the balance between purple and green has been messed up. You'll block it going in, but the hydrogen will still be flowing out, and that will end up with more red pluses. You see in B, you have more red pluses on the outside. The cells are messed up, they are not growing, they're not happy at all. And so they mutated the TRK channel. But what they did is then they, into the mutant what's messed up and not growing, they added the amphotericin B, and the amphotericin B allowed them to replace the potassium transport that was mutated away. Again, we're like number A again. The amphotericin B by itself could replace the potassium channel that was needed for the wild type. This is kind of a cool experiment. You mutate the potassium channel away, then you replace it with this small molecule that makes its own potassium channel that allows the cell to grow and to thrive. So the thing about the amphotericin B molecules is they make a ring, and it's got a potassium-sized hole in the middle. In fact, we've got the structure of that ring figured out right now. I want to say this is a lipid that's doing what we showed you the glucose transporter was doing. More about lipids when we get to them. I just want to say lipids can do what proteins were doing in the other case. It's exactly what lipids are doing here. It's all about making a hole in the membrane, a polar hole that will be the right size to allow the ion through. So all that is to say, we've got all these things that we're talking about. Just remember that these are in the terms of the enzyme kinetics terms that we're talking about. Simple diffusion, facilitated diffusion, is the one that's being catalyzed. Simple diffusion is the uncatalyzed analogy. Then you have the things where you can do active transport, which means you're burning ATP, and this will be something where usually you're burning ATP for a reason. That means you're moving the substrate probably through a positive delta G reaction, but you're paying for it with ATP's negative delta G. You can also have secondary active transport, where you have one of the two things that's going on can be negative delta G, and it can pay for a positive delta G transport, or maybe they're both negative. Maybe they won't both be positive, because then it wouldn't happen. You have ion channels, which are passive. The important thing about ion channels, they're passive and they'll allow things to move down their gradient when they are opened up. And then you have ionophores, which act like ion channels, but they are chemicals that are concentration dependent, and they will mess things up. They will kind of be a shortcut through the membrane, which will undo all the hard work the sodium-potassium ATPase has been doing. Okay, so that was putting together the two things. I'm going to do one more short video that returns to Chapter 6 and does the two enzymes, but I think I will keep all this together.